Okay, so today we're going to go through Krebs cycle and the electron transport system. So we're still continuing with the oxidation of glucose. Next time we'll talk about metabolism of other fuel sources like proteins and lipids. But right now we're still focusing on the oxidation of glucose. And we went over glycolysis last time and then what we ended up with at the end of glycolysis. So this image here just kind of is a good quick review as to where these three processes take place. So here's the plasma membrane of a cell. Remember that glucose needs to pass through the plasma membrane via a glucose transporter, gets inside of the cell, and then again, we're following the oxidation of glucose just to make ATP. So you start with glucose, you go through all of the steps of glycolysis. At the end of glycolysis, you end up with pyruvate, a little bit of ATP, and then you also end up with 2 NADH plus H plus, 2 of your electron carriers getting reduced. This does show you end up with the right amount of ATP. This here, remember you end up with 2 pyruvate when you start with 1 glucose. And then what happens to pyruvate, again, assuming oxygen is available, and oxygen is not always available. We'll talk about what happens to pyruvate if oxygen is not available. But assuming it is, the pyruvate needs to get moved into the matrix of the mitochondria. As it passes across the membrane of the mitochondria, pyruvate gets converted to acetate. Coenzyme A attaches to it. And then acetyl coenzyme A is what is going to enter the Krebs cycle, all of the steps of the Krebs cycle. Krebs cycle is also called tricarboxylic acid cycle. You may have learned it as that, the TSA cycle. It's also called the citric acid cycle. So you go through all of the steps of the Krebs cycle. In the process of that, you're going to reduce some more electron carriers. You're going to reduce some more NADH plus H plus. You're going to make a little bit of ATP. You're also going to reduce your other electron carrier, FADH2. And then the carbons that entered the cycle, you are going to lose as CO2. Now, all of the electron carriers that have been reduced, they are going to now, in the mitochondria, they're going to transfer their electrons to the members of the electron transport system. You have transfer of electrons from one member to the next until finally oxygen accepts the electrons. And in the process of doing that, you can make a lot more ATP. So this just quickly reviews the anatomy of the mitochondria. Here's the outer membrane. Here's the inner membrane. Krebs cycle takes place inside of the inner membrane, which is the matrix. The members of the electron transport system are embedded into this inner membrane. It's also called the cristae. And the space between the inner membrane and the outer membrane is called the intermembrane space. That space is important in terms of creating a hydrogen ion concentration gradient. Okay, so this is where we left off last time. This was our final slide for glycolysis. Again, for glycolysis, you start with 1 mole of glucose. At the end of all of the steps, you end up with 2 pyruvate. Remember, pyruvate and pyruvic acid are the same thing. 2 ATP and 2 NADH plus H plus. And just to remind you again, the difference between the oxidized and reduced version of your electron carriers. You must have the oxidized version of your electron carriers available. If you do not have those available, then your reactions of glycolysis will stop at 3-pgal. Because in the next step, you have to pull electrons off of 3-pgal, transfer them to NAD plus to reduce it to NADH plus H plus. So if you do not have the oxidized form of this electron carrier, these reactions cannot continue. And at this point, you have invested 2 ATP, but you haven't made any ATP. So the cell would actually be consuming ATP and not producing any. So what happens after glycolysis? What we're going to focus on is what happens when oxygen is available, but we still need to talk about what happens when there is no oxygen available. So if oxygen is available, again, that's called aerobic metabolism, then the pyruvate that's produced will be converted to acetyl-CoA, and then that is what will enter the Krebs cycle. Through the reactions of the Krebs cycle, you'll produce more electron carriers, a little bit more ATP. The NADH plus H plus that was produced at the end of glycolysis will carry the electrons into the mitochondria and transfer them to the electron transport system. NADH plus H plus transfers its electrons to the electron transport system, and then now this gets oxidized. So now you have regenerated the oxidized version of your electron carrier. This can move freely back and forth across mitochondrial membranes, so it can move into the cytoplasm where it can be used again in glycolysis. Without oxygen, so that means that the cell has run out of oxygen, but cells have to have a way to continue to make ATP because in order to keep themselves alive, they need ATP. So there has to be some mechanism to make ATP until the oxygen supply can be replenished to a cell. So what happens when oxygen is not available? When oxygen is not available, the pyruvate that is produced will end up getting reduced to lactic acid. The reason why pyruvate gets reduced to lactic acid is because you have to have a way to regenerate the oxidized form of your electron carrier. And you have reduced an electron carrier. With oxygen available, this will be pulled into the mitochondria and the electrons will be donated to the electron transport system. When there isn't any oxygen available, you don't have that pulling the electrons into the mitochondria. So those electrons have to get transferred to something else. So again, the cell must oxidize its electron carrier so it can continue to make ATP. So this is just a little bit more detail here. With oxygen, again, this is aerobic metabolism. Pyruvic acid is produced at the end of glycolysis when oxygen is available. Pyruvic acid is an acid, and this is important. Even if you call it pyruvate, it's an acid. And so acids can't be allowed to remain in the cytoplasm because if you keep an acid in the cytoplasm, it's going to cause the cytoplasm to become acidic. Now, again, if oxygen is available, then the pyruvic acid is going to get moved out of the cytoplasm. It's going to cross the mitochondrial membranes. And as it does that, it gets converted to acetyl. Coenzyme A attaches to it. And then that is what helps to facilitate the addition of acetyl into the Krebs cycle. So by moving the pyruvic acid out of the cytoplasm into the mitochondria, you have prevented the acid from affecting the pH of the cell. So the pH remains constant. So that's with oxygen. Without oxygen, your end product is lactic acid. So when you do not have oxygen available, the final thing that accepts the electrons that have been moved around is pyruvic acid. It gets reduced to lactic acid. But again, lactic acid cannot be allowed to stay in the cytoplasm because it would cause the cytoplasm to become acidic. So what happens is that your liver is the only tissue in your body that can actually clear lactic acid. I'll talk about what that means in just a minute. So you still have to have a way to get this lactic acid out of the cytoplasm. So it gets moved out of the cell, it gets moved into your blood, and there it gets transported to your liver. And in your liver can take lactic acid and process it. So again, the pH of the cytoplasm remains constant. It remains unchanged. So this is what happens to the electrons and pyruvic acid when oxygen is not available. So for anaerobic metabolism, again, you have to have a way to regenerate the oxidized version of your electron carrier. And so remember, at the end of glycolysis, you produce pyruvate, produce 2 pyruvate and 2 NADH plus H plus. Now, if you don't have any oxygen available, you have to have a way to regenerate this. So these electrons get transferred to pyruvate. Pyruvate gets reduced, NADH plus H plus gets oxidized. So now you've regenerated the oxidized form. This is pyruvate, which is a 3-carbon intermediate. And here is pyruvate once you've added the pair of electrons. And that is going to give you lactate or lactic acid. So again, that lactic acid gets moved into your blood and gets transported to your liver. So this is showing you a muscle cell, this is showing you your circulatory system, and this is showing you your liver. So the series of reactions that is going to result in lactic acid getting converted to something else is called the Cori cycle, and this happens in your liver. So this is just showing you a muscle cell. Any cell in the human body has the ability to go anaerobic. Some tissues can go anaerobic more quickly and for maybe a little bit longer than other cells. Some tissues can't go anaerobic at all, and we'll talk about that again in just a minute. So it's showing you muscle because muscle uses a lot of ATP, and skeletal muscle frequently goes anaerobic. It's a normal part of its actions throughout the day. So this is just showing you glycolysis here. So you start with glucose, you end up with pyruvate and a little bit of ATP, again, to regenerate your electron carrier. The electrons get transferred to pyruvate, converts it to lactate or lactic acid. That gets moved into your blood and shipped off to your liver. Your liver has the enzymes to convert lactate or lactic acid back to pyruvate. And if you remember, let me go back over here. All of the reactions of glycolysis are reversible. So the enzyme that will convert 3-phosphoglyceric acid to 2-phosphoglyceric acid can also be run in reverse. And that's what happens in the liver, is it takes all of the reactions of glycolysis and runs them in reverse. But more importantly, it has this enzyme right here to convert lactate or lactic acid to pyruvic acid. Runs the reactions in reverse to make glucose. Now there's some terms down here that we need to go over. And what I just described, the process of taking pyruvate and running the reactions of glycolysis in reverse to make glucose, that's called gluconeogenesis. Gluconeogenesis, gluco is glucose, neo is to make new. So this is making glucose. And what's important here is it makes free glucose. What that means is that it's actually just glucose. And your liver is the only tissue that does this. And the reason why is because glucose can readily move across cell membranes via glucose transporters. So once the liver has taken the lactic acid, converted it to pyruvic acid, run the reactions in reverse to make glucose, then a couple of things can happen. If glucose levels in your blood have been depleted because tissues all of a sudden increased their metabolic activity, used a lot of the glucose available, if blood sugar levels have gone down, then some of this glucose that's made by the liver needs to get moved into your blood to restore blood sugar levels. If blood sugar levels are okay, or once they have been restored, then any remaining lactic acid that gets converted to glucose can then get used to make glycogen. Okay. Glycogen is a cell's storage form of glucose. Cells don't have glucose just hanging around because it'll get run through Krebs cycle. So if a cell doesn't need to use glucose to make ATP, that extra glucose is used to make glycogen. So it has a reserve of glucose in the cell. Glycogen is a very large macromolecule. It cannot move across the cell membrane. So your liver can make glycogen, and skeletal muscle is another tissue that will commonly make glycogen. So glycogenesis is making glycogen. And again, that's going to happen in your liver and skeletal muscle. And then if a cell all of a sudden increases its cellular metabolism and it needs to tap into that extra store of glucose, it can take glycogen and convert glycogen to glucose. Except when a cell does this, it doesn't go from glycogen back to glucose. It typically converts it to fructose 6-phosphate. So it doesn't run the reactions all the way in reverse. And the reason for that is because, again, if skeletal muscle took glycogen and converted it back to glucose, that could leave the cell. Because now all of a sudden you'd have a higher concentration of glucose inside the cell because you've broken it down from glycogen than outside, and glucose could leave the cell. You don't want that to happen. So if you stop at fructose 6-phosphate, this isn't going to get across the plasma membrane. It has this large negatively charged phosphate group attached to it. So glycogenolysis, glycogen is using glycogen. Lysis is to break down or break a bond. So you're breaking the bonds of glycogen to use the glucose that is produced. Now again, in the liver, it can run the reactions all the way in reverse to make free glucose, or it can run the reactions in reverse to make fructose 6-phosphate and then use that for its own cellular metabolism to make ATP. Both skeletal muscle and your liver have a high metabolic requirement. They need to make a lot of ATP. So you want to make sure that you know these three terms and where they can happen and the details of the Cori cycle. So why would cells switch to anaerobic metabolism? They would switch to anaerobic metabolism basically when they run out of oxygen. And again, for some tissues, this is a normal part of their daily activity. They can switch back and forth between aerobic and anaerobic metabolism quite frequently. What does happen though, if a cell runs out of oxygen and it has to shift to glycolysis only as a method of making ATP, then you go into what's called oxygen debt. So oxygen debt is the amount of oxygen that is required to restore blood sugar levels, to restore your glucose levels in your plasma to what they were originally before these tissues went anaerobic, to restore glycogen reserves in liver and skeletal muscle. So once that has all been returned to its normal set point values, the amount of oxygen that was consumed to do that is called the oxygen debt. Again, some tissues are better adapted to anaerobic conditions than others. Some cells in your body, glycolysis is the only mechanism of making ATP, and red blood cells fall into that category. I think we mentioned earlier that, human red blood cells are anucleate. In fact, they've lost all of their organelles once they have produced a sufficient amount of hemoglobin. So if red blood cells don't have any organelles, they don't have mitochondria, and this is where the reactions of Krebs cycle and electron transport system take place, which is where oxygen is used to make ATP. Red blood cells don't have that, so the only way they can make ATP is via the lactic acid pathway or by anaerobic metabolism. Again, other tissues that will frequently switch between aerobic and anaerobic metabolism is skeletal muscle. Bone marrow can do this, peripheral nerves, and in the renal medulla, so the middle part of your kidney tissue. So these are some tissues that will commonly switch back and forth between anaerobic metabolism, but that doesn't mean that all cells in your body can do this as readily. There are a couple of notable exceptions, and that is your heart and your brain. The muscle cells of your heart, your myocardial cells, have no ability to go anaerobic, and same with neurons in your brain. So these tissues have very little or no ability to go anaerobic. And so if your overall cellular metabolism increases in your body, you'll get something called glucose sparing. And what glucose sparing does, let me write it on here for you. Glucose sparing is where you have some tissues of the body that will choose to not use glucose to spare it for other tissues that have no ability to go anaerobic, and they spare it for them. And so that's called glucose sparing. So glucose sparing, again, is where you have cells that will use other fuel sources instead of glucose to spare it for some tissues like the heart and the brain. Okay, so now we need to continue on with aerobic metabolism. So again, if oxygen is available, the pyruvic acid that's produced at the end of glycolysis, again, is an acid, so it can't remain in the cytoplasm. It has to get moved out of the cytoplasm because it could affect the pH and cause the cytoplasm to become acidic. We've already talked about that. That can't happen. So what happens is this next step in the process is called the intermediate step. The pyruvic acid gets moved into the mitochondria, and as it passes across the outer membrane of the mitochondria, it gets converted to acetyl coenzyme A. And again, this is called the intermediate step. So remember, at the end of glycolysis, when you start with 1 mole of glucose, you make 2 pyruvic acid or 2 pyruvate. Glucose is a 6-carbon intermediate, pyruvic acid is a 3-carbon intermediate. So you have 2 of these. So as pyruvic acid crosses the outer membrane of the mitochondria, it's going to lose a carbon and get converted from a 3-carbon intermediate to a 2-carbon intermediate. This 2-carbon intermediate is called acetate. So anytime you lose a carbon from something, you lose it as CO2. So this is where CO2 is produced from cellular metabolism. This is one of the places. The other thing that happens is you remove carbon and oxygen to CO2, but you'll also transfer another pair of electrons from pyruvate to NAD plus to reduce it to NADH plus H plus. So you've reduced another electron carrier. And then the coenzyme A attaches to acetate, and coenzyme A is the coenzyme for the enzyme that is going to be involved in adding acetate to the intermediate of the Krebs cycle to start the cycle. So acetyl-CoA then will continue on through the Krebs cycle. And remember, this is produced inside of the mitochondria. So Krebs cycle is another series of metabolic reactions that's involved in the metabolism of carbohydrates, proteins, and lipids. So right now we're just focusing on the oxidation of glucose. Next time we'll talk about the metabolism of proteins and lipids, and we'll see how the Krebs cycle plays a role in the metabolism of those other fuel sources as well. But again, right now we're going to focus on just glucose. So we talked about glycolysis being an anaerobic process because it does not require any oxygen. Krebs cycle technically is an anaerobic process because none of the reactions of the Krebs cycle require oxygen. However, the reactions of the Krebs cycle cannot continue in the absence of oxygen. Remember, this is happening in the mitochondria, and again, you have to have a way to regenerate your oxidized versions of your electron carriers. And so the electron carriers are going to transfer their electrons to the electron transport system. And what is driving that entire process or pulling those electrons towards the electron transport system is oxygen. So even though Krebs cycle itself does not require oxygen, it cannot run without oxygen present. And so what we're going to do is go through the steps of Krebs cycle, just like we did for glycolysis. And for every turn of the Krebs cycle, meaning for every acetate that enters the cycle, you're going to make 1 ATP. You are going to reduce 3 NAD plus to NADH plus H plus, and you're also going to reduce 1 FADH2. So you're utilizing another electron carrier here as well. So again, you have to remember that at the end of glycolysis, you make 2 pyruvate. So if we're calculating the number of ATP that is made when you oxidize glucose, you have to calculate this as 2 turns of the Krebs cycle. And then at the end of Krebs cycle, an electron transport system, glucose is fully oxidized to CO2 and water. So this is the Krebs cycle. Again, this is showing you what you get for 1 turn. 1 turn of the Krebs cycle is where you have an acetate entering the cycle, and you go through all of the sequential reactions of the Krebs cycle until you get to the point where you produce your sort of your starting metabolic intermediate, the one that's going to combine with a new acetate that enters the cycle. That's 1 turn of the Krebs cycle. And again, in 1 turn, you're going to make 1, 2, 3 NADH plus H plus. You're going to make 1 FADH2, and you're going to make 1 ATP. So again, if we're focusing on glucose, then that's going to be 2 turns of the Krebs cycle because you have 2 pyruvate. Each pyruvate gets converted to acetyl-CoA. Okay, so this is a cycle, which means technically you can start any place in a cycle, and you should be able to get all the way back to that same exact intermediate. And that's what you find when you're talking about metabolizing other fuel sources like amino acids or lipids. So remember that your pyruvate, as it crosses the outer membrane of the mitochondria, you lose a carbon as CO2, so it gets converted to this 2-carbon intermediate, which is acetate, and then coenzyme A attaches to it. So that acetate is going to combine with oxaloacetate. So oxaloacetate plus acetate gets converted to citrate. Now, coenzyme A is the enzyme involved in facilitating the enzyme here in this reaction. So you have oxaloacetate, you have to add in a water molecule to combine it with the acetate to give you citrate. Oxaloacetate is a 4-carbon intermediate, acetate is a 2-carbon intermediate. So you have 4 carbons plus 2 carbons, that gives you citrate, which is a 6-carbon intermediate. In the next step of the Krebs cycle, citrate gets converted to isocitrate. So in this reaction, the bonds just get rearranged. You don't add anything, you don't lose anything. In the next step, isocitrate gets converted to alpha ketoglutarate. two things happen in this step. You lose a carbon, a CO2. So isocitrate is a 6-carbon intermediate, alpha ketoglutarate is a 5-carbon intermediate. You lose that carbon, a CO2. You also will reduce one of your electron carriers, NAD plus to NADH plus H plus. So by doing those two things, alpha ketoglutarate, that converts isocitrate to alpha ketoglutarate. In the next step, you have 3 things that happen. No, 2 things. No, 3 things, I'm right, sorry. Don't mind me. Alpha ketoglutarate gets converted to succinyl coenzyme A. So here's where you lose that 2nd carbon that was added into the cycle as CO2. You reduce another electron carrier. So the electrons get transferred from alpha ketoglutarate to NAD plus to reduce it to NADH plus H plus. You remove carbon as CO2. And then here you have to add in coenzyme A again. So alpha ketoglutarate gets converted to succinyl coenzyme A. Now, in the next step, succinyl coenzyme A gets converted to succinate, which means the coenzyme comes off. And remember that enzymes can be used over and over again. So you have a pool of coenzyme A that's hanging out in mitochondria. Now, what happens in this step is you have to pull in a phosphate, and this is coming from just the pool of inorganic phosphate in the matrix of the mitochondria. So you go from succinyl coenzyme A, you add in a phosphate group, you're going to lose the coenzyme, and that phosphate group gets attached to succinyl. So you attach the, to go from succinyl coenzyme A to succinate, you add in a phosphate group in this step, and that phosphate group is combined with GDP to make GTP, and then the phosphate group is pulled off of GTP and added to ADP to make ATP. So 1 ATP is made, and you have an enzyme that's pulling that phosphate off of a substrate and transferring it to ADP to make ATP. That substrate is GTP. So here, initially, you have an inorganic phosphate that's just coming from the pool of phosphate groups that's hanging out inside of the mitochondria. So you get a free phosphate group attached to GDP to make GTP, and then you can pull the phosphate off of GTP, add it to ADP to make ATP. Again, the coenzyme comes off, and you've converted succinyl coenzyme A to succinate. In the next reaction, succinate gets converted to fumarate. So it's going to get oxidized. It's going to lose a pair of electrons. The electrons that come off of succinate are going to get transferred to FAD plus to reduce it to FADH2. So now you've produced fumarate. In the next step, fumarate gets converted to malate. You have to add in a water molecule to do that. And then in the very last step, you go from malate to oxaloacetate. So malate gets oxidized to oxaloacetate. You pull a pair of electrons off of this, transfer them to NAD plus to reduce it to NADH plus H plus. Now you have regenerated oxaloacetate, and now the reactions can continue, and you can add 1 more acetate into the cycle, and these reactions can continue. So again, for 1 turn of the Krebs cycle, you're going to reduce 3 electron carriers that are NAD plus. So here's 1, 2, 3 of those. You make 1 ATP, and you reduce 1 FAD plus to FADH2. So again, if we're tracking the amount of ATP that's produced from the oxidation of glucose, you're going to have to count 2 turns of the Krebs cycle. So again, in order to regenerate your starting product, which is this 4-carbon intermediate, you initially add 2 carbons into the cycle to give you a 6-carbon intermediate. You rearrange the bonds, still have a 6-carbon intermediate. You lose 1 carbon as CO2, that takes you down to a 5-carbon intermediate. Then you lose the 2nd carbon that you've added in as CO2. Now you're back down to a 4-carbon intermediate, and everything past this point now is still your 4-carbon intermediate. So what you need to know from this is the sequence of steps. You need to know that acetate combines with oxaloacetate to make citrate. And the only way that acetate can be entered into this cycle is if it is facilitated and transported by coenzyme A. Once the acetate enters the cycle, the coenzyme is released and can be used to facilitate this reaction again. It's the same coenzyme that's down here. So you just have a pool of coenzyme A that is hanging out in the matrix of the mitochondria that can be used in the Krebs cycle. So know the sequence, the names of the intermediates, and what happens at each step. You're not responsible for knowing the structure, just know what happens at each step. So before we go through what happens next, which would be the electron transport system, I have to show you or talk a little bit about how, like, logistically where these reactions take place. So this here represents the inner membrane of the mitochondria. So that means this here, which is where Krebs cycle takes place, this is the matrix. This is the intermembrane space, because out here you'll have your outer membrane of the mitochondria. So the components of the electron transport system are embedded into this inner membrane of the mitochondria. You have electrons that are going to be transferred to the electron transport system, coming from NADH plus H plus and FADH2. So this step right here, where succinate gets oxidized to fumarate, you pull a pair of electrons off of succinate, they get transferred to FAD to reduce it to FADH2. This is happening in the matrix of the mitochondria, and this is complex 2, which has iron in it, and this is where the electrons are going to get transferred from FADH2 to the iron of complex 2, and then from there it gets transferred to another component, which is called coenzyme Q. That's then going to transfer the electrons to complex 3. The electrons that are being transferred from NADH plus H plus are going to get donated to one of the members of complex 1. But complex 2, each of these complexes has a name based on basically the first enzyme involved in what's happening in that complex. And so the name of complex 2 is succinate dehydrogenase. A dehydrogenase is an enzyme that's going to dehydrogenate something. It's going to pull a pair of electrons off of something. Oops, sorry about that. It's going to pull a pair of electrons off of a substrate and transfer them to something else. That is a dehydrogenase. So this is an enzyme that you need to make sure you know to recognize its name and what it does. So a dehydrogenase is going to remove 2 hydrogens from a substrate and transfer them to another molecule. That's a dehydrogenase. So succinate dehydrogenase is telling you that it's going to pull a pair of hydrogen ions from succinate. They get pulled off of succinate and they have to get transferred to something else. They get transferred to FAD plus to reduce it to FADH2. That's this enzyme that does this, and that's actually part of complex 2. So here's succinate dehydrogenase here. So I wanted to go over that to show you how these metabolic pathways, where they're located and why the location of them is so important. So the components of complex 1 and complex 2 that accept the electrons are again going to face the matrix. So the electrons that are going to be transferred from NADH plus H plus are going to get transferred to FMN, which is part of complex 1. The electrons that are getting transferred by FADH2 are going to get transferred first to iron of complex 2. And then from either complex 1 or complex 2, the electrons are going to get transferred to complex 3, then complex 4, and then finally to oxygen. So again, this is the reactions of Krebs cycle, which we just talked about. Now it's turned about 90 degrees to the right. When we go through the reactions of Krebs cycle, we start with acetate here, combining with oxaloacetate. So it's the exact same sequence of reactions. It's just showing you the starting point here at like 3 o'clock instead of noon. Okay, so there's a lot of detail of the electron transport system. So I'll show you a diagram, go through the detail, and then you have the diagram again. So when you're studying for this, you want to make sure that you can talk your way through the entire process. If you can do that from start to finish without looking at any notes, then you have a really good understanding of the process. So again, sometimes it's called the electron transport chain, which is why the C is here instead of an S. The electron transport system is a series of complexes. There are 4 of them involved in oxidation reduction reactions. They're going to transfer a pair of electrons from one complex to the other. Remember that electrons are transferred as pairs, and they're going to first get transported to these complexes, and then they're going to be carried between the complexes by electron carriers. One of them is coenzyme Q, the other one is called cytochrome C. Now, transferring these electrons is coupled to pumping protons or hydrogen ions from the matrix to the intermembrane space. So 3 of the 4 complexes have a proton pump associated with it. Each of the complexes are embedded into the inner membrane of the mitochondria, and the very last thing that accepts the electrons is oxygen. So the final electron acceptor when oxygen is available is oxygen. And when oxygen accepts 2 electrons, remember we're following them as hydrogen ions, then oxygen combines with hydrogen to make water. What is driving this entire process to pull the electrons through these different components of the electron system, and what's driving the pumps, is the electronegativity of oxygen. That proton pump, those pumps that create this hydrogen ion concentration gradient, that concentration gradient is used to make ATP. And again, this entire process is driven by the oxidative power of oxygen. It's very electronegative. It has a very strong pull on those electrons. So here's my version of the electron transport system. There are a lot of different diagrams of the electron transport system, and they're all a little bit different in terms of the detail that they focus on. This is the detail that I want you to know at this point. So if you take a cell and molecular class, or if you take a biochemistry class, you'll go through electron transport system again, and you'll talk about some additional details of it. So you're going to be required to know more detail than you did when you first learned it in your intro bio course. But again, when you take other courses, you'll focus on some different details, a little bit different than, or in addition to what we're going to go over. So this is the entire process. Here are your 4 complexes. Here's complex 1, complex 2, complex 3, complex 4. Again, the complexes also are named based on the first enzyme involved in the reactions that take place. A complex means it's made up of multiple subunits. So complex 1 is called NADH dehydrogenase. Complex 2 is called succinate dehydrogenase. Complex 3 is cytochrome reductase. Complex 4 is cytochrome oxidase. You need to know these names. Again, here is the inner membrane of your mitochondria. That means this space here is the matrix. This space here is the intermembrane space. Here's your outer membrane of the mitochondria. So the reactions of Krebs cycle are taking place here. So electrons that are going to get transferred to the electron transport system, that are being transferred by NADH plus H plus, are going to transfer those electrons at complex 1. Now, it doesn't matter where NADH plus H plus comes from. It doesn't matter if it's coming from the reactions of Krebs cycle, which are happening here, or if it comes from the NADH plus H plus that is produced during the intermediate step, or if this is the NADH plus H plus that was produced by glycolysis. It doesn't matter where it comes from. If the electrons are being transferred by NADH plus H plus, they get transferred to FMN, which is one of the components of complex 1. So NADH dehydrogenase, it's going to dehydrogen this. So it's going to pull a pair of hydrogen ions off of NADH plus H plus. That will oxidize it to NAD plus. The electrons get transferred to FMN. Now, this is important because right here, you've regenerated the oxidized version of your electron carrier. And the same thing applies. If you don't have the oxidized version of this, the reactions of Krebs cycle can't continue. And if you can't continue through the process, you can't make any ATP, and you can't reduce electron carriers and transfer them to this system, which is where you're going to make a lot more ATP. So when oxygen is present, this is how you regenerate the oxidized version of your electron carrier. So again, the electrons get transferred from NADH plus H plus to FMN. FMN is then going to transfer the electrons to an electron carrier, which means it's going to carry the electrons to the next component of the electron transport system, or the next complex. So the electrons get transferred from FMN to coenzyme Q. Coenzyme Q can move laterally through the membrane. Coenzyme Q accepts the electrons, so FMN gets oxidized, coenzyme Q gets reduced. Coenzyme Q then shuttles the electrons over to iron of complex 3. I just realized I forgot to add arrows here. It first goes to iron of complex 3, and then from iron of complex 3, the electrons get transferred to cytochrome C, which is then going to shuttle the electrons from complex 3 to complex 4. Once the electrons get transferred to complex 4, they go from cytochrome C to cytochrome A3. This is just one member of this complex. And from cytochrome A3, the electrons get transferred to oxygen. Oxygen combines with the hydrogen ions to form water. So you have two electron shuttles in the electron transport system, coenzyme Q and cytochrome C. Both of these are going to move between these complexes and transfer the electrons to the next complex. So electrons that are donated by NADH plus H plus go to FM1 of complex 2 to coenzyme Q. This then shuttles them to complex 3. Iron accepts them, so iron gets reduced to Fe2 plus. Then they get transferred from iron to cytochrome C. That's going to oxidize iron back to Fe3 plus. Cytochrome C gets reduced. They get transferred from cytochrome C, cytochrome A3, cytochrome A3 to oxygen. Oxygen gets reduced when you add the hydrogen ions or electrons to water. Now, if the electrons are getting donated to this system by FADH2, remember these electrons that were used to reduce FAD to FADH2 were pulled off of succinate. So this enzyme here is succinate dehydrogenase. It's going to pull the hydrogen ions from succinate, transfer them to FAD plus to reduce it to FADH2. FADH2 is then going to transfer the electrons to iron, which is a component of complex 2. From iron, they get transferred to coenzyme Q. It's just another copy of this. From coenzyme Q, they get transferred to iron of complex 3. Iron of complex 3 gets reduced to Fe2 plus. From there, they get transferred to cytochrome C. Cytochrome C then will transfer them to cytochrome A3 of complex 4. From cytochrome A3, the electrons get transferred or accepted by oxygen to reduce oxygen to water. So again, you have electrons coming in and being entered into or transferred into this system, so they can then be transferred from one complex to the next complex to the next complex. So here's where the electrons enter the electron transport system. The very last thing that accepts the electrons is oxygen. And when oxygen accepts the electrons, it gets reduced to water. And again, oxygen has a very strong oxidative power. It has a very strong pull on these electrons because it's so electronegative. So it's literally like a magnet that is pulling these electrons through the complexes and ultimately towards oxygen. Now, as these electrons get transferred from complex 1 to coenzyme Q, and from iron of complex 3 to cytochrome 3, and from cytochrome A3 to oxygen, every time they get transferred from a complex to the next component, that is going to power this proton pump. So these three complexes have the proton pump associated with them. And what they do is they're going to pump hydrogen ions from the matrix into the intermembrane space. So what happens is you create a high concentration of protons or hydrogen ions in the intermembrane space. Now that concentration gradient of protons is going to be used to make ATP. So we haven't talked about ATP production yet. We've just been talking about the electron transfer that is going to create this concentration gradient of protons. That concentration gradient is then used to make ATP. So I apologize, I forgot to put the arrows here first, but just keep in mind they go from coenzyme Q to iron of complex 3, and then from iron of complex 3 to cytochrome C. It doesn't matter where coenzyme Q is shuttling the electrons from, whether it's complex 1 or complex 2. So it's important to note here that iron is a component of these two cytochrome complexes. And iron, again, is something that is stable in both its oxidized and reduced forms. So about 1/4 of the iron in your body is used to make up these two complexes of your electron transport system. So of the total amount of iron in your body, half of it is used in hemoglobin, 1/4 of it is used in your cytochrome complexes, and then the remaining 25% is stored in your liver. So you have iron if you need it. So the next couple of slides just go through the entire process that I just went through. So you can go back and read this. And again, you should be able to talk your way through this without having to look at the notes. So again, this just goes through what I just said, describing the system. And then here it is again. So you go over it one time with the diagram, you read through the transcript of what happens at each step, and then hopefully you can go through this diagram again, and the second time you go through it, it would make a little bit more sense. Okay, so that's the electron transport system. So again, we still haven't talked about the ATP that's made from the electrons getting moved through this electron transport system. So these 4 complexes are the complexes of the electron transport system. Now there is an enzyme. It's a hydrogen ion channel which has enzyme activity that's also embedded into the membrane of the mitochondria. And that is called ATP synthase. So that inner membrane is going to have a hydrogen ion channel. So the hydrogen ions that were pumped across the membrane to create this high concentration of hydrogen ions can move through that hydrogen ion channel from this intermembrane space back down into the matrix, because it's going from an area of high concentration to an area of low concentration. As those hydrogen ions pass down their concentration gradient, there's a little bit of energy that's released. And that free energy that's released as those hydrogen ions pass down their concentration gradient is used to attach a phosphate to ADP to make ATP. So the phosphorylation of ADP is coupled to oxidation, because oxidation is driving this entire process to create that concentration gradient that is then used to make ATP. So that process is called chemiosmosis. Chemiosmosis is where you couple oxidation to phosphorylation. And again, what's driving this entire process is the electronegativity of oxygen. That is what is pulling the electrons through the electron transport system complexes. 3 of those complexes have pumps associated with them to create that gradient. So ATP synthase is the enzyme that's going to catalyze the synthesis of ATP from ADP and inorganic phosphate. Now, the phosphate that is used to attach to ADP is coming from the pool of phosphate ions or phosphate groups that are just hanging out in the matrix. So the phosphate group isn't coming off of a substrate and getting transferred to ADP. The concentration gradient is used to pull a phosphate group from the matrix and attach it to ADP. So this is the enzyme involved in that. So this shows you right here here is where that hydrogen ion channel that has the enzyme activity is located. So again, here's complex 1, complex 2, complex 3, complex 4. Complex 1, 3, and 4 have the proton pumps associated with them. So every time electrons get transferred, the pump is powered to pump hydrogen ions from the matrix to the intermembrane space. That creates a high concentration of hydrogen ions in the intermembrane space. And then in that membrane is this hydrogen ion channel, so they can freely pass through this channel from an area of high concentration to an area of low concentration. As they do that, as they fall down their concentration gradient, you get a little bit of free energy that's used, that gets released. And that free energy that is released is captured and used to power this enzyme to pull a phosphate group from the matrix, attach it to ADP to make ATP. This is chemiosmosis. This cannot happen without oxygen pulling the electrons through this electron transport system and creating this hydrogen ion concentration gradient. So through glycolysis, Krebs cycle, and electron transport system, we've actually talked about two types of phosphorylation. In glycolysis and Krebs cycle, you have what's called substrate level phosphorylation. The phosphate group that is used to attach to ADP is pulled off of a substrate. So ADP is phosphorylated to make ATP. It's still this exact same reaction. In substrate level phosphorylation, this phosphate group comes off of an intermediate. The enzyme pulls it off of an intermediate, attaches it to ADP to make ATP. So the phosphate group is directly transferred from a substrate by an enzyme to make ATP. Enzymes that are going to transfer phosphate groups to or from ATP that are transferring this phosphate group are called kinases. Okay, so remember we talked about kinases initially as pulling a phosphate group off of a substrate, attaching it to ADP to make ATP. We talked about that with glycolysis. Those kinases can be used in the reverse reaction as well. So these are kinases. So in glycolysis, this happens in two places. It happens as you go from 1,3-biphosphoglyceric acid to 3-phosphoglyceric acid, which is 3-pgal. That's where you make your first ATP. Here you have two phosphates. You pull a phosphate off of this, attach it to ADP. That gives you an intermediate that only has one phosphate group. And then the last place where ATP is made is the step that goes between PEP, or phosphoenol pyruvate, to pyruvate. Phosphoenol pyruvate is pyruvate with a phosphate group. You pull the phosphate group off of this, transfer it to ADP to make ATP, and that gives you pyruvate. So those are the two places where substrate level phosphorylation happens in glycolysis. And in Krebs cycle, there's only one step where you make ATP. And initially, remember, the phosphate group is pulled out of the matrix and added to GDP to make GTP. And then the phosphate group is pulled off of GTP, added to ADP to make ATP. So this is substrate level phosphorylation because the phosphate group that's used to add to ADP comes off of GTP. That's substrate level phosphorylation. The process that we just talked about with the electron transport system is oxidative phosphorylation, because phosphorylation is tied to oxidation. So remember that concentration gradient of hydrogen ions is created by the oxidizing power of oxygen. And then that concentration gradient is used to capture energy to attach a phosphate group, which is just in the matrix of the mitochondria, to ADP to make ATP. That's oxidative phosphorylation, and that's what happens in the electron transport system. So again, we have followed glucose through glycolysis, the intermediate step, and Krebs cycle to make ATP. So how much ATP have we made when oxygen is available? We know how much ATP you make when oxygen is not available. If oxygen is not available, then glycolysis is the only pathway that is available to make ATP. At the end of the process, you make 2 ATP directly. Now remember, if oxygen is available, you still have your electron carriers. So your 2 NADH plus H plus are going to take these electrons, move them into the matrix of the mitochondria, donate them to FM1 of complex 1. From the intermediate step, you've also reduced another electron carrier, right? Now remember, you have 2 pyruvate that are going to cross into the matrix. So you have 2 NADH plus H plus. And then the acetate that was converted from pyruvate in that intermediate step is going to enter the Krebs cycle. For every acetate that enters the Krebs cycle, you make 1 ATP. You reduce 3 NADH plus H plus and 1 FADH2. So you've already made a little bit of ATP from substrate level phosphorylation. And remember, these numbers here are multiplied times 2 because you have 2 pyruvate. But these electrons that are coming into the electron transport system are going to be transferred to the electron transport system ultimately to make ATP. So oxidative phosphorylation takes the energy transfer of the electrons to make ATP. And how much ATP is made? When the electrons are transferred by NADH plus H plus, I forgot to add the plus here, you get a total of 3 ATP that are made. When the electrons are transferred by FADH2, you make 2 ATP. So you have 3 ATP for each pair of electrons coming from glycolysis, 2 times 3 ATP here, because you have 2 pyruvates that are moving into the mitochondria. And then 2 times 3 is 6 more NADH plus H plus here, times 3 ATP. And then you have 2 ATP made from each FADH2 that's produced from the 2 turns of the Krebs cycle. So you can do some simple math and determine how much ATP you make with oxygen, right? Without oxygen, all you make is your 2 ATP from glycolysis, okay? So that's substrate level phosphorylation. The intermediate step, you don't make any ATP. In glycolysis, you make 2 ATP and you reduce 2 electron carriers. You make 2 NADH plus H plus. Intermediate step, you do not make any ADP. You're going to reduce 2 more NADH plus H plus because there's 2 pyruvate produced at the end of glycolysis. So each pyruvate is going to get moved into the mitochondria. That's going to give you 2 turns of the Krebs cycle. Each turn of the Krebs cycle makes 1 ATP. So 2 turns of the Krebs cycle, you make 2 more ATP. Again, this is substrate phosphorylation. But for each turn, you make 3 NADH plus H plus and 1 FADH2. 2 turns of the Krebs cycle means 6 NADH plus H plus and 2 FADH2. So how much ATP are you making from these electrons that are transferred to the electron transport system? Again, anytime electrons are donated by NADH plus H plus, you make 3 ATP. So 2 times 3 gives you 6 ATP. These are the electrons that have been pulled from intermediates in glycolysis. When these electrons get transferred to the electron transport system, you oxidize this, you regenerate your oxidized electron carrier, it can go back and be used again. The 2 electrons that are transferred to the electron transport system by NADH plus H plus from the intermediate step, again, each pair of electrons that are donated by NADH plus H plus, you make 3 ATP. So 2 times 3 gives you 6 ATP here. You have a total of 6 NADH plus H plus that are produced from your 2 turns of the Krebs cycle. 6 times 3 gives you 18 ATP. You have 2 FADH2 that are produced from the Krebs cycle. For every pair of electrons that are donated by FADH2, you make 2 ATP. So 2 times 2 is 4. So if you add up all the ATP that was made, you made a total of 4 ATP by substrate level phosphorylation. Again, that's coming from glycolysis and Krebs cycle. And then from oxidative phosphorylation, I spelled that wrong. Phosphorylation, you make a total of 34 ATP. So 34 ATP by oxidative phosphorylation plus your 4 ATP from substrate level phosphorylation gives you a total of 38 ATP. So when you start with 1 mole of glucose, when oxygen is available, you make 38 ATP. You start with 1 mole of glucose and you have no oxygen available, you make a total of 2 ATP. So you can see that with oxygen, you can make a lot more ATP. And this is why we are aerobic organisms. as an entire organism, we have such a high metabolic requirement. We need so much ATP that we could not possibly survive on the amount of ATP that's made from anaerobic metabolism alone. So what you want to do is you want to make sure that you can follow all of this, right? One of the things that you need to keep in mind is at the end of glycolysis, you have 2 pyruvate. So this is going to happen twice. This is going to happen twice. Okay. And then calculate these out so you know how much ATP is made. Now, again, it's important to remember what you get from the Krebs cycle, from 1 turn of the Krebs cycle. Let me go back to this really quickly. We can even go right here. From 1 turn of the Krebs cycle, so this is from 1 acetate that enters the Krebs cycle, you make 1 ATP, 3 NADH plus H plus, and 1 FADH2. The reason why you need to memorize these numbers is because next time we're going to talk about the metabolism of amino acids and lipids. And those are going to enter into the Krebs cycle at different locations. So for every acetate that enters the Krebs cycle, you're going to produce this. And especially when you are metabolizing your lipids, lipids are going to get broken down into many acetates. And then each one of those acetates will be run through the Krebs cycle. So we'll see how much more ATP you can make when you metabolize lipids. When you metabolize amino acids, which come from proteins, you make about the same amount of ATP as you do from glucose. But if you metabolize lipids, you make a lot more ATP. And that's because they generate a lot of acetates, and so you get many more turns of the Krebs cycle. Amino acids are what enter the Krebs cycle in different locations. Let me go back to Krebs cycle and show you. So each of these intermediates, your different amino acids can get converted to these intermediates. And so amino acids will enter the Krebs cycle at different locations. And they basically go through 1 turn of the Krebs, which is why you get about the same amount of ATP that's produced from an amino acid than you do from glucose. But if you metabolize lipids, lipids get chopped up into many little tiny acetates, and every acetate that's produced is going to be 1 turn of the Krebs cycle. So for example, a relatively small amino acid, you may have 20 turns of the Krebs cycle from a relatively small fatty acid or triglyceride. So 20 turns of the Krebs cycle is a lot more than the 2 turns you get from glycolysis. So you want to make sure you know the terms of chemiosmosis. An additional enzyme that you need to know the name of is a dehydrogenase. You need to know that that is going to dehydrogen something. So it's going to pull a pair of hydrogen ions from a substrate and transfer them to your electron carrier. So that's how you reduce your electron carriers, is by dehydrogenases. Okay, and that's it for Krebs cycle and the electron transport system. Again, next time we'll talk about the metabolism of proteins and lipids.